Hello everyone, and good afternoon. It's been 1 week I have not had a class. Okay, I hope that you are doing well. So thank you very much for joining today's class. Yeah, I was busy last week because I was marking the Form 5 papers. Okay, we were rushing for the deadline, and also wasn't feeling very well actually. Okay, I'm also not very well today. I hope I don't start coughing afterwards. Okay, anyway, so I will try to manage today's lesson. Yeah. Okay, so today I'm starting chapter 11. Okay. Let me see. I think I see Kenneth here. Good afternoon, Kenneth. And Muhammad. Okay, Muhammad, who's around at he messaged me at 7:29 a.m. Okay, good afternoon, everyone. So I hope that you're all ready for today's class. So today's class will be a bit short, I think, but I don't want to have it too much because I may not be able to handle such a long time, and then I may start coughing afterwards, and then there'll be a disaster. That'll be disastrous. Okay, let me start sharing screen. Okay, now today we're going to do immunity in humans. Actually, it's quite short, right? It's not very difficult to handle. It's very simple. You just need to understand a few concepts. Okay, if you have any questions, as usual, please do put your questions here. All right, and I should be happy to answer them. Okay, let's look at immunity, right, in humans. Okay, first 11.1. Okay, body defense. So here the picture, I'm sure you will recognize this is a virus that is being under attack by this white blood cell. Okay, so you should be able to recognize with all these spikes. Okay, this is one of the common shapes of the virus, which is spherical, round, with a lot of these projections, which are spikes here, that help to stick onto the cell so that they can attack the cell. Okay, let me start. Right. Now, body defense. Okay, first of all, there are a few terms that you need to remember, that you need to know. Okay, when does our body defense start? When are we triggered? When is the body defense triggered? Now, it is triggered, or it is stimulated, or is it initiated? Okay, so you can use this word interchangeably. Okay, triggered, that means started. Stimulated also can. You can use initiated also can. When pathogens infect the body. Pathogens are substances like bacteria and virus and anything that can give us disease. They are called pathogens. So basically, it's a general term. Okay, so pathogens are microorganisms that causes diseases. So it's not good for us, right? Pathogens. Under pathogen, we normally will have these groups of microorganisms called bacteria, virus, protozoa, and also one more is fungi. Okay, all these four can cause diseases. So not algae, even though algae is microorganisms, but they are not known to infect and cause diseases to other cells. Okay, so look at these examples. So here, the first picture here is a bacteria, all right? I don't know why it mixed with the virus there. Okay, the one on the right-hand side, that is virus. Then the one at the bottom, these are protozoas, some examples of protozoas. And these are fungus that are cultured on a petri dish. Okay, examples, bacteria, virus, protozoa, and fungi or fungus. Okay, so these are pathogens. So they can find their way into our system if we have what we call our membrane has already been broken, or our what they call the first line of defense has there is a way for them to come in, then we will have a problem. All right, our body will be under attack by this bacteria and virus. Okay, so pathogens can only cause diseases if they successfully infect body cells. So if they do not enter our body cells and they do not infect us, in fact, pathogens are perfectly fine. We don't fall sick. If you have a pathogen in front of you, if it's on the table, you may have a virus in front of you. Okay, on the table here, bacteria. You're not going to be sick unless it comes into your body. Okay, so the most important thing is we need to prevent these bacteria or these pathogens from entering our body. So our defenses are very important. Okay, the first thing is to try to prevent them from entering, and if it enters, then we will go to the second mode of defense, which is to try to fight them. Okay, so the first is always prevention. Prevention is the best cure. Okay, like people say. Okay, let's go to the next one. Right. So what are antigens? So if we talk about this chapter, you cannot run away from knowing these terms like antigen, antibody, immune response, immunity, and so on. Okay, so what are antigens? So antigens is actually any foreign particle or any molecule, all right, that the cell, our body cells, recognize as something foreign. Foreign means it doesn't belong to our body. It is not supposed to be here inside our body. So usually these are protein molecules. Okay, protein molecules, they have a certain structure, they have a certain shape. So if they are not part of our body, our cells should be able to recognize that this should not be here. Okay, especially our white blood cells. This will trigger our white blood cells to produce a response, which we call the immune response, to try to kill or get rid of these foreign things. Okay, so antigens are foreign particles that enter the body and stimulate our white blood cells to react to it. So there must be antigen before our bodies produce the response. Okay, because this antigen is the trigger. When it enters our body, it will trigger our white blood cells to go into action, to start to fight it. Okay, so here you will see these are antigens, right? They could be in the air, they could be in the dust, okay? It could be in the wherever it's all around us. So if we manage to inhale them into our body through our nose, through our airway, or through any part of our body, or through the wound, okay, then we will have a war in our hands. Okay, that means your body cells will try to fight these invaders. Okay, so antigens, these are antigens, actually the protein, all right, or the substance that is on the surface of these foreign particles. These antigens will stimulate the lymphocytes. The lymphocyte is one of the white blood cells, one type of the white blood cells that are specialized to produce a chemical called antibody. So this chemical, the function is to destroy the antigen. So only when the antigens come in, that's when the antibodies will be produced. Antibodies usually are not produced before there is presence of antigen. So antigen is the one that stimulates or induces our body to produce that antibody. Okay, so you can see here one of the reactions are, besides producing antibody, is to actually to kill off the bacteria by trying to eat up the bacteria. Okay, now let's carry on. Now let's look at the antibody molecule. So usually this antibody molecule will be represented by a Y shape. There's a figure there, okay? Because this is what the antibody molecules look like. You have long chain and you have a short chain, okay? And they basically look like that, all right? So antibodies are proteins. So remember that, antibodies are actually protein, just like antigen also. The antigen is foreign protein that should not belong to our body. Okay, but antibodies are proteins that are made inside our body. The white blood cells, particularly lymphocytes, make these antibodies. Okay, and they are released into the blood when there is antigen. Okay, so this is another picture of the antibody. So the interaction between the antibodies and the antigen. So when the antigens come in, so our body will produce that antibody, and then they will react. Okay, the interaction, this is called the immune response. So you have immune response if you are able to produce antibody. Okay, if you are not able to produce antibody, that means you don't have an immune response. You are not able to protect yourself. Okay, you will definitely fall sick if you do not have this immune response. Okay, and this is a picture of a virus, okay, where you have this surrounded by antibody. So what the antibody does is actually is try to stick onto these projections, all these protein spikes that you have on the surface of the virus or bacteria, so that they will minimize the harm, because all these spikes are supposed to attack or hold on to the membrane of your cells, okay, your body cells. So if they're able to hold on to it or to grip onto it, that's when they will start to attack your cells by injecting their substance, which is the nucleic acid, into the cell, and that your cell is destroyed. Okay, and you will make your cell make more copies of the virus. Okay, next. Okay, let's look at the next one. Now, what is the meaning of immunity? So you have seen antigen, you've seen the meaning of antibody. You also know what is immune response, the interaction between antigen and antibody is the immune response. Now the immunity. The immunity means the body's ability to fight infections. So if you do not have the ability to fight infections, it means you do not have the immunity. Okay, when you don't have the immunity, it means you will fall sick. So it's the ability to fight infections caused by pathogens through specific reaction to pathogens. Okay, so the immunity is good because that will prevent you from falling sick. Okay, so this is a picture of immunity. When you are immune, when you have immunity, it's like physically you're able to block out, okay, you are able to stop this virus from making you fall sick. Okay, so when our body can resist a disease, we are said to be immune. Okay, and we are not immune to all diseases. Okay, otherwise we will not fall sick. So sometimes there are certain diseases we are immune to, some we are not able to immune. That's why sometimes we get infected, we fall sick, and then we need additional help. Okay, that's why we take in antibiotic. Antibiotic are these drugs. Okay, they are made by fungus, help our body to kill this bacteria. So we need reinforcement. We need help from outside. Okay, if we already fall sick. Okay, now let's look at lines of defense. Okay, this is more the central part of this chapter is to know there are 3 lines of defense. Okay, when you have an enemy attacking your country, all right. So the first line of defense will be normally situated at the border, right? When it comes in through the enemy at the border, all right. So this is the first line of defense, and they will try to stop it, try to stop this invasion. If that doesn't happen, if it's not able to stop the invasion, then you have other defenses that will be the backup. Okay, let's look at the 3 lines of defense. There are 3. First line of defense, this refers to physical and chemical barriers. Okay, our body has got physical barrier. Barrier is like a protection. It's like a membrane. Something that covers, something that blocks is called barrier. Then we have chemical substances that our body produces, like tears, sweat, and inside there it produces, and there are enzymes here that can have the effect of killing the bacteria. Okay, so everything that's physical and chemical barrier, that comes under the first line of defense. Okay, now, if the first line of defense is breach means the first line is unsuccessful in blocking out the enemies, or we call the pathogen, then we will have another line of defense that will be, will go into action. That will be the second line of defense. The second line of defense happens when the first line of defense is not able to block out. So this bacteria and virus has gone into the blood system. Okay, has entered our body now. Okay, this is the general responses to infection, including eating up the bacteria. So we have this process called phagocytosis. Okay, eating up the bacteria and so on. Okay, we will explain that further. And if that also fails, right, if that is not enough to kill off the bacteria, we have our third line of defense, which is very, very specific, which is very, very specialized. Okay, so this is where we have our antibody. Antibody will be produced when the this bacteria enters our body and is not able to be killed off by the second line of defense. So now the third line of defense is we will have no other choice. We will produce antibody to kill off this antigen. Okay, so we have 3 lines of defense. All right, now what comes under the first line, second line, and third line? You should know that. That is going to be asked in your exams. Okay, this is the central part of the chapter. The entire chapter, you should know all these 3 things. Okay, here. First line of defense consists of physical and chemical barriers. So think of your body. What kind of physical barrier do you have? What kind of chemical barriers do you have? Okay, maybe I should like to ask Kenneth. Can you give me some example of some physical barriers or chemical barriers? Either one you can choose. What's a physical barrier? Any part of our body is able to block out bacteria or not, or pathogen or not. And if you can't think of any physical, think of any chemical that our body produces that can kill bacteria or that can minimize the effect of these pathogens. Yes, Kenneth, do you have any? I think your teacher would have taught that in school, right? I think, right? I think I should think that I'm the slowest, right? I'm quite slower because I had some other times I could not have class, so my lessons would be a bit slower compared to what your schools are teaching actually. All right. Okay, Kenneth. Okay, no response. Maybe a bit slow. All right. Okay, never mind. I will explain now, right? Consists of physical and chemical barriers. We will have things like this. This is all over your body, right? You have your skin, which is a very, very good physical barrier. Then you have blood clotting. Okay, this is a stomach, right? Your stomach has got acid inside there, which is your hydrochloric acid. This is a chemical thing. So now you have, the function is to prevent pathogens from entering the body. Okay, so you have things like this, right, sorry. Later I'll go through the detail, I think. Okay, so let me go through this again. Non-specific response. This is important. This is a non-specific response. What does that mean by a non-specific response? It means that these physical and chemical barriers that you have under the first line of defense does not distinguish what bacteria, what virus that comes in, and it should be able to have a general defense. That means this line of defense should be able to block out all bacteria and all virus. There's no specific, this type, I must use this defense, okay, or this barrier. This barrier is only for this bacteria, or this barrier is only for that virus. No, so it's general. This is a general barrier that is able, should be able to stop the invasion or prevent the pathogens from coming into the body. So it's non-specific. It does not have to specify. This virus needs this type. That virus needs another type. Okay, that's what it means. Okay, so let's go to first one, skin. You can see under magnification, you can see our hair is quite tough, right? I mean, it's quite coarse. You can see all the pores there. It provides a physical barrier. So the skin is a physical barrier that is impenetrable. Impenetrable means not able to be passed by, okay, or pass through. Pathogen cannot pass through. If your skin is not cut, your skin is not injured, no wounds, it should not be able to allow entry of any of these pathogens. Okay, that means your skin is all right now, right? It's not clear, it's not tampered with, it is not injured, right? Then it should be all right. Okay. It is also a chemical barrier. Okay, besides being a physical barrier, it's also a chemical barrier because our skin produces certain chemicals. Okay, you may be not aware of it, right? Sometimes our skin has some kind of an oil. That oil is called sebum. Okay, creates secretes sebum. So in the follicle, the hair follicle, right, just beside it there is a sebaceous gland. Sebaceous gland is the gland that produces some kind of oil. So this oil, besides lubricating the skin, all right, preventing wrinkles and so on, it also has a protective function. It contains lactic acid and oils. So it will be able to prevent, it will attack some of the, it will prevent or maybe even kill off some of the bacteria, or can break down the cell wall of the bacteria. In the sweat, okay, it also contains the lysozyme. This is an enzyme which is found in the tears, also found in the sweat, and it's also the function is to break down the cell wall. Okay, so our skin will have chemical barrier. It's a chemical barrier. There is sebum, there is sweat that contains lysozyme. Okay, next. Mucous membrane. Okay, our lining of our throat, the what you call the passageway, our air passageway, our intestinal glands and all that, the lining, there is a layer of membrane, all right, which is called the mucous membrane. So why do we call it mucous membrane? A membrane is just a layer, right? Mucus because it is able to produce mucus, okay? And mucus is actually a good thing. Okay, so all these mucous membrane, they line the inside of the trachea, the respiratory passageway, the bronchial, and so on. Digestive system also, all along our esophagus, okay, the duodenum, and so on. And also the urinary tract, which is the ureter, okay, and the urethra. We also have the mucous membrane there. The function here is to produce certain substances that are called mucus. Okay, especially when in your throat here, sometimes you find your phlegm is a little bit thick because you have this mucus. So it has already trapped a lot of dust. Okay, so when you breathe in dust as well. And you can also have bacteria and so on. So it goes in together with the air. So the function of this mucus is to trap bacteria. So this is a physical barrier. All right, in order to trap the bacteria, then it's all stuck in the mucus. What do you do with the mucus? So sometimes you throw it away, right? You spit out your mucus. Sometimes you're not able to spit out, you will swallow it. When you swallow it, it will go into your stomach. Okay, and that's fine. Okay, that's fine. It will not go into your throat, it will go into your stomach because in your stomach there is hydrochloric acid. It will have it can have the function of killing the whatever is in the mucus, the bacteria that you happen to trap together. Okay, so in the nose, there is also hair, right? You can see that nose hair. A lot of people, especially men, they have a lot of nose hair. So they also serve a function to trap, to separate the pollutants from the air, and also once it gets these dust particles get trapped in the mucus, we will also bring it down, okay, and then if you swallow it, it will be killed off in the acid juices, the hydrochloric acid. Okay, you can see all this hair. These are the cilia. You can see here. I think it's not very clear here. I will just draw up here. So these are the cells, all right, that have cilia. And these special cells are called goblet cells, which are able to produce these mucus. So you can see all these goblet cells, they produce mucus. So they are your respiratory tract and all these digestion, they are never dry. You have a lot of these substances helping to trap everything that comes together in the air or whatever, okay, that enters your body. So these are under the first line of defense. Okay, mucus contains lysozyme. So the same chemical that we saw just now, it is able to break down the cell wall of certain bacteria. Okay, lysozyme. Okay, so now we go to the specific lysozyme. They are found in many parts of the body that's liquid, right? Like tears, saliva, nasal mucus. Nasal mucus is the mucus that comes from our nose. So normally when we cry, right, we have a lot of mucus production. Then sometimes when we are sick, when we have flu, the mucus production will be increased. Okay, and also sweat. So all these have this chemical called lysozyme. And they are antimicrobial. That means they are able to kill, okay, they are able to kill microorganisms. These are proteins that can break down the wall of the microorganisms, able to destroy bacteria, okay, in other words. All right, okay, so this is very clear cut, very simple also, but you just need to remember what is first line, what is second line, and what is third line. You need to know how to categorize them. Okay, all this so far is first line. Okay, still some more, two more. Blood clotting. Blood clotting has the function of plugging the hole. Okay, when you have an injury, your blood will flow out, blood cells and so on. So when you have blood clotting, it actually plugs the wound there, and it also prevents the entry of bacteria. So this is one of the functions of blood clotting, which you've seen earlier in the previous chapter. Okay, and also hydrochloric acid. This one we mentioned, when you swallow your mucus, you swallow your phlegm, it goes into your stomach, and there is this hydrochloric acid that helps to break down whatever bacteria that happens to be in your mucus there. Okay, so they are produced by the gastric glands in the stomach. Okay, so this is very simple. Nothing you need to go into detail. Just need to know how to name them. You just need to name them. Okay, and leaf-lice bacteria in the food as well. Okay, so let's go to the second line. Okay, so now if the first line of defense has been breached, that means it's not effective enough to block out the bacteria that or the microorganism that happens to be very strong, right? It doesn't get killed off, or maybe there's a wound. When there's a wound, of course, it will go into the blood, all right, this bacteria. So now it's up to your second line of defense to carry on this war to protect your body. Okay, So secondly, what comes under the second line of defense? You have three things here: phagocytosis, fever, and inflammation. Okay, let's look at one by one. Okay, let's look at this as a phagocytosis. This is general. Later on, I'll explain in detail. So I'm sure you have seen this word phagocytosis when you learn about, remember amoeba? Then when you learn about the life cycle of amoeba, how they eat. So you've seen that they engulf the food. They will bring in the food into the cell. Then it forms a vacuum. And then the lysosome will have enzymes. It will break down the food, digest it, and absorb the nutrients. Okay, so the same principle also, but this is carried out by the white blood cells in our body cells, in our body, okay? So you have phagocytosis, trying to consume, this process of consuming the bacteria, eating up the bacteria and killing it in the same time. Then you have fever. Fever is also a second line of defense. It serves a purpose also. Why your body temperature rises, it serves a purpose. And inflammation, okay? Inflammation is when your injury site, the place where you got injured, it becomes red and becomes what you call swollen. It's painful, okay? Because it is like, it becomes a bit bigger, right? And it bulges out, and there's a lot of pain there, and it becomes red. That's what it means by inflammation. Okay, next. So this second line of defense is also non-specific. That means it does not specify. Okay, this kind of virus, I must use this method to overcome it. Okay, there's no such thing. Any bacteria, any virus will also meet the same thing. Will also meet the phagocytosis. Okay, you have all these reaction. It's the same reaction for all the bacteria and all the virus that comes into your body. Okay, next one. Let's look at phagocytosis. Yeah, you have to memorize all the line of defense. Yes, correct. Because they will ask you, explain, describe the first line of defense, the mechanism of first line of defense. So you're able to talk about the lysozyme, right? Lysozyme, then what you have, your physical barrier, what are the considered physical barrier, what are the chemical barriers, your sebum, your skin, and blah, blah, blah. So you need to know what's under the first. Okay, so the second, right? So you go to the second now. Phagocytosis. So if you want to explain, okay, let's say a very simple question is, they show you a cell eating up another cell, and they label that as white blood cell, not amoeba. If you don't label, of course it could be amoeba, right? So you see a white blood cell eating up your this one. So let's say the question, explain the mechanism that this cell uses to destroy the bacteria. That means you are supposed to explain phagocytosis, the mechanism, the process. So here, this is also can be a question. So what is the definition first? Phagocytosis is a process of trapping and digesting microorganisms by phagocyte. Now, phagocyte is the cell that is able to carry out phagocytosis is called a phagocyte. So you see a lot of terms. Actually, if you understand the meaning of this word phagocyte, then you will more or less can. Cyte means is the cell. Phago is to eat. Okay, so a phagocyte is the cell that eats up other cells. And the process is phagocytosis. Okay, so the phagocyte is referring to the cell. So example, I'm looking at a phagocyte now. This is a phagocyte because it's carrying out the process of eating up the whatever bacteria. Okay, I have another question here. What's the best way to memorize all the biology facts? Okay, the best way is to read. Best way to understand first. Then you read as much as you can. You read and make short notes, and use the acronyms. You know acronyms? Sometimes you have a list of terms. You use the first letter and you make a sentence out of it. Okay, like rainbow. I know the colors of rainbow. You have ROYGBIV. How to remember? Red, orange, yellow, green, blue, indigo, violet. Green, blue, indigo, violet. Because I make this acronym called ROY-GB. Roy, R-O-Y, Roy. G is the G for green. B-I-V, beef. It's like somebody's name. So try to think of what's the ingenious way of memorizing. Yeah, you have to. Wherever I can, I will give you lah. Okay, like after I give you one, I got one acronym for you to remember. Okay, so You can message me later if you need any more help in trying to remember certain things. Okay, let's go on with this one. Now, involving, if you want to talk about the mechanism, 5 steps. Attraction. What's the mean attraction? Now, the pathogen, the phagocyte must be able to know that the bacteria is there. So it has to be attracted to it. So you have to move towards it. Attraction is able to recognize there's a presence here and is somehow being attracted. So this phagocyte will move towards this bacteria. Okay, so it's attraction. Next, you have ingestion. The next stage is to try to bring it inside. Ingestion is actually the name eating. When you say ingest, do not ingest, that means do not eat. Okay, when you have a packaging that is actually maybe a preservative or something, or is to absorb the oxygen and say, do not ingest, that means do not eat lah, that means eat. Ingestion means eating. So this bacteria, this phagocyte will extend the pseudopodium, all right, this one now, where all this, and surround it, engulf it, and bring it into the cell. So that process is called ingestion. Right, next. After ingestion, you'll find that there's going to be a vacuum. Vacuum, you see this vacuum? This vacuum. It can be called a food vacuum, okay, if it happens to be a food lah. So here, this vacuum is called a phagolysosome. At first, it's called phagosome first. Now, phagosome means this vacuum contains the bacteria. Okay, whatever that's eaten is called phagosome. Now, afterwards, it forms a phagolysosome. And why do you change the name from phagosome to phagolysosome? A bit more complicated. Because phagosome just contains the thing that is the cell has just swallowed, right? Has just engulfed. But when you talk about phagolysosome, is you see this one, this is the lysosome. Lysosome contains the enzyme. Once it merges, see, it merges together. The lysosome merge with the phagosome. You call the entire thing as phagolysosome. Okay, so now it's called phagolysosome when the lysosome merge, or emerge together, join together with the phagosome. All right, okay, so after that, what is the purpose of merging? Because the lysosome has enzyme. So the enzyme has the function of breaking down. It has the lysis kind of the effect. It's going to break down. It's going to make the cell burst, right? So this is called digestion. So lysosomal enzymes, that means enzymes that are able to break down. It's called lysozyme. It will digest and break down the material that is already inside, captured already. Okay, and lastly, after breaking down, right, all these substances will be removed out of the cell. Okay, we call it ejected. Okay, so we have this word called egestion. Okay, not ejected, but egestion. Egest means to remove it out of the cell or remove it entirely out of the system. They are released from the cell. Okay, so remember attraction, ingestion, then the formation of the phagolysosome, after that digestion, and then egestion. Egestion is the opposite of ingestion. They are just opposites of each other. Ingest is to makan, bring inside. Egest is to push it out. Okay, that's what it means. Yeah, all right. So phagocytosis, now this is a diagram in the textbook. Okay, you can see this, the same thing in the textbook. It has the same effect, the same facts here. So it extends its pseudopodium. This is the attraction. Okay, after attraction, the cell has to push, extend the pseudopodium in order to cover it, to engulf it. We call it envelops the bacteria. Then it forms the phagosome. Then the phagosome combines with lysosome. You get the phagolysosome, this one. After that, this lysozyme that is inside here, inside the phagolysosome, will break down the bacteria. And then whatever that the cell may need, it can be absorbed. Whatever the cell does not need, it will be expelled. So this is ingestion. Okay, you should be able to explain the whole system, the whole process. Then you can get full marks actually. It's not difficult. Okay, So another one is a fever. Why do we often, when we fall sick, why do we have fever? Okay, So there is a function here. So when you have a higher body temperature, when you have a fever, this makes the phagocytic activity, that means the process of eating up, become more active. So you're making your phagocytes become more active, eating up the bacteria if your temperature is higher. Okay, so at the same time, this high temperature also helps to denaturalize the protein of the pathogen, because we mentioned the antigens, they are actually protein also. So if you have a higher temperature, it might be able to denaturalize the protein of the pathogen. Okay, and then this is denaturation, showing the process of where the bonds that holds up the shape of the protein is broken. Then you find the shape already, already altered, changed. And then fever also stimulates increase in the production of antibodies. So antibody production may also be even faster if the temperature of the body is a bit higher. So it has a function to help to aid the process of defending your body. All right, okay. Next, right. Inflammation. Inflammation when the part becomes swollen. I'm sure you have seen this before, right? It becomes bengkak, and maybe called bengkak, or becomes swollen, and usually associated with red color as well. Okay, the body, your inflammation is actually your body's normal response to the injury. Whenever you have an injury, you will find that it becomes bengkak, right? It becomes swollen. It is your body's response that makes the thing become red, that makes the thing become bigger. Okay, so let's look at this diagram. Look here, the person has been, pierced by a little bit of spike here. So you will see that the blood vessels, one of the responses here is, you find that the blood vessels will dilate. This is to increase more of the red blood cells, the white blood cells to flow there. And you also see the white blood cells coming out of the blood vessel, the capillary. It will squeeze through these pores, okay, that makes up the capillary wall. So it squeezes out. These are the white blood cells in order to help to kill whatever bacteria that comes in. Okay, so this area will swell, it will become red and become painful. This is your reaction to the injury. That's why it becomes red. Okay, so this is a picture from the textbook. Now you see the damaged tissue. Whenever you have an injury, the damaged tissue produces a histamine. Histamine is a chemical that is produced whenever there's an injury. Okay, and this histamine is like a signal. It's like calling for help. It's asking your body to come to its aid. Okay, so this histamine will cause the blood capillary to expand. So when it expands, more white blood cells will flow there, and also red blood cells, of course, right? So it will make the white blood cells go there, and it will diffuse out into the injury site, so that it's able to kill bacteria in case bacteria comes in. Okay, so histamine will cause the blood capillaries to expand. That's one of the action. It also increases the permeability of blood capillaries to phagocyte. It allows this white blood cell to move out, okay, of the blood capillary, so that it can go to the injury site. And the phagocytes and clotting factors will also accumulate here, and the blood clotting mechanism will be triggered, will be started. That one you've learned earlier in your chapter 10. Okay, so this is another word here, histamine. Histamine is the substance that is produced by at the injury site. That is like a signal calling for help, that all these things will come to its aid. All right, okay. So third line of defense, let's say this is not efficient enough to kill off your bacteria. So your third line of defense will be activated. Okay, your third line of defense is what we call the antibody antigen, or we call the immune response. So this is specific. Okay, so the first one and the second line of defense is not specific. So it is general for all bacteria and all the virus that happen to come in, for all the pathogen. But when it comes to the third line of defense, you need specific antibody to overcome certain kind of antigen. This has to be a specific response. Not one antibody can act on all the antigen. You have antibody, you have antigen A, you need antibody A to act on it, okay, or to bind to it. When you have antigen B, you need another antibody, which is antibody B, in order to bind to it. Okay, so when a pathogen enters the body, let's say the second line of defense is not efficient enough, not effective enough to kill off, then the antigen on the pathogen will stimulate your lymphocyte. So lymphocyte is a specific group of the white blood cell. White blood cell, as you know, there are many types. There are 5. So this lymphocyte is one of them, and the function is to produce antibodies. Okay, so now the action of antibodies are specific. Please remember to write this whenever you write answers for essay. When you talk about third line of defense, when you talk about antibody, you must use the word specific. Antibody reactions are very specific. There is no one antibody that can act on all types of antigen. A certain antibody is needed for certain type of antigen. Okay, right. So let's look at here. Now, I'm sure some of you may have experienced this before, right? You feel, you're feeling under the weather, you're not feeling well. Sometimes you happen to touch your neck and then you see, but suddenly something is like, bloated up here, satu biji like that, one like a nut, right? A bongo here in your neck somewhere. You feel it and then you start to fear. Okay, am I having cancer or what? Right? Is this what they call a tumor? Okay, sometimes you don't have to be so alarmed. It just happens to be a lymph node that is swollen. Look at the difference in the size. This is a swollen lymph node. This shows that this person is undergoing some kind of an infection. Okay, there's bacteria in the body at that site, somewhere near the head or the nose or the mouth or the throat. And this, there's a lot of activity in the lymph node. The white blood cells are being actively produced. Okay, macrophages are there. They are killing off bacteria at that time. And this, the green one, is the normal lymph node that is not swollen. Okay, so the lymphocytes may accumulate in the lymph nodes and cause the lymph nodes to swell, to become swollen, become bigger, engorged. So, and the lymph nodes also contain macrophages. Now, maybe you see this new word. Macrophages are just white blood cells that are specialized. They have already undergone some kind of a change to become very efficient at eating up bacteria. So they're actually neutrophils and monocytes. Neutrophils and monocytes, when they need to carry out phagocytosis, sometimes they can become bigger. They can change to become macrophages, and that happens in the lymph nodes. So they are very efficient at doing their job, which is to destroy bacteria and kill bacteria through phagocytosis. So macrophages' job is just one: eat up bacteria. Okay, it's like just sucking up, vacuuming everything, killing off all these bacteria that are not supposed to be there. Okay, so they are actually, before that, they're actually monocytes and neutrophils. All right, okay, so you see monocyte here, a monocyte here on the left hand side, it has become specialized, changed into become a macrophage, which is more suited to carry out the job of eating up the bacteria, phagocytosis. Okay, next. So lymphocytes are divided into 2 types actually. Okay, so I'm sure you've seen the T cells and B cells. You may have heard, right? So what are the differences between these 2 types of cells? T cells, their function is to attack cells that are already infected by pathogen. Okay, T cell. So this, why we call T cell? Because these cells were made in the bone marrow, but they migrate to the thymus when they are young, right? When they are when it's after they are being formed, they will migrate to the thymus to mature. So they call it T lymphocytes or T cells. Okay, so T cells, they attack infected cells. Then what about the B cells or the B lymphocytes? The B lymphocytes, they are the ones that produces memory cells. The T lymphocytes stimulate B lymphocyte to produce memory cells. So memory cells are these cells that are able to recognize the antigen. If in the future, all right, this person gets infected by the same bacteria or the same antigen. So these memory cells are already there. They recognize this is the type of antigen that was in here before. So they are immediately activated to produce antibody. And this causes the person to have immediate immune to this disease. So the person does not fall sick the second time. Okay, so you have memory cells. Memory cells, once they are made, they stays in your body. It stays in your body to protect you from another infection. Okay, that's why you do not get certain diseases 2 times, like measles, or this we call it chicken pox. You don't normally get it twice because you have memory cells inside there. Okay, so the B cell activation, it can activate it to become memory B cells, which will be able to recognize the antigen. So if the same pathogen attacks, the memory cells will be stimulated to produce antibodies immediately. So that means there's no lapse, there's no gap, there's no waiting time. Because if there's a waiting time, that's when you will fall sick. If there's no waiting time, immediately if the antibody is there, it kills off your pathogen. Okay, now, so you can see this word, the specificity of antibody-antigen reaction. So you can see that the Y shape here, that I'm talking about this antibody, whenever you look at this Y shape, it refers to your antibody. And you can see that at the one of this end, already you have this long chain and the short chain here, the antigen, it also, the receptor site is very specific, just like when you learn your enzyme and your substrate, okay, the kind of reaction where you have the lock and key kind of thing. So it's the same thing here. So you will see that so this explains why you need certain antibody for certain antigen. Because the shape, you see the shape, each type of antibody can only combine with a certain type of antigen. So therefore, you cannot have one antibody that can fit all types of antigen because of the specific shape. Okay, so this one can only fit the orange color one. You can see because the shape fits. The green one cannot fit here because the configuration is different. Okay, so the antigen molecule combines with the antigen receptor site. So there's a receptor site on the antibody molecule. It is able to combine because the shape fits. So the configuration, this is the correct one. Okay, remember that. So this is what we're looking for when we mark your paper, to see your facts, whether you are able to use these words or not. Specific, okay, antibody, antigen response, receptor. All this will give you marks, okay, rather than just beating about the bush and using normal words, a layman term, learn terms that people don't learn biology can use. Okay, so you have to show that you have the fact by using the correct term, and that's where we give you the mark. Sometimes we are very specific. We want the word, specific word. If you don't say active site for the enzyme, you don't get your mark. You have to use the specific words. Okay, now the last part. This is going to be a short one. Actions of antibody. So we know the antibody will bind with the antigen. What's going to happen to your antigen after that? Okay, what's going to happen to your antigen? You know the antibody is supposed to do something there. Okay, what can happen to your antigen? So there are a few things that can happen. There are 5 things that can happen. Either one of them. These are the reaction. You have agglutination. There is neutralization. There is precipitation. There is opsonisation. And there's lysis. Okay, now I said, Muhammad asked me how to remember. Okay, try to remember, use acronym. Okay, NAPOL, let's say NAPOL. Okay, you can put LOPAN, LOPAN also can. Depends on you, right? N is for neutralization, A is for agglutination, P, precipitation, O, opsonization, and L for lysis. Okay, let's look at it one by one. So agglutination, okay, this is a simple way to remember after I tell you how to remember this. Antibodies will coagulate. You've seen it before when you talk about the blood, different blood group being donated into, transfused into a different non-compatible blood. You notice that they will stick together, right? They will form clumps. They will clump together. We have this process called agglutination. So the same thing, antibody can also coagulate the pathogens, make them stick together. Okay, so make them easy targets to be tracked. So they stick together, so they are not so mobile. So they're not so mobile, they cannot do the harm. They cannot harm your host cells. They cannot harm your cells because they are not able to be very mobile. And how to kill off? They will be destroyed by pathogens. Phagocytes. They'll be destroyed by phagocytes. So later on, these macrophages will come. The macrophages will come and then they will eat up everything. So that's how they are killed off. Okay, so you need to agglutinate them. That means you need to stick them together, and then your macrophages comes. Okay, so you see this is a diagram, right? Generally, if you see all of them sticking together, like forming a kind of network, that means agglutination. So how to remember? Look at this word glue, G-L-U, glue. Glue means stick, isn't it? Stick together. So that helps you to remember. Glue means stick together. Okay, next, neutralization. Okay, neutralization, as you know, when something is neutralized, that means it's not able to have the original function. Okay, so these antibodies will bind with toxins. Usually it's referring to toxin. Toxins are poisons or what you call proteins that happen to destroy your cells and so on. So these toxins are produced by bacteria. So the antibody will bind with the toxin. So you can see here, they also fit a specific shape, right? Your toxin has a specific, let's say this is a wedge shape, then your antibody will just fit right into it. And what it does is it neutralizes the toxin. Okay, neutralizes, use the correct word. If it's neutralization, you use the word neutralize. Okay, and then how to remember? You see the word T here, neutralize the toxin. Okay, neutralize the toxin. So it will make the toxin unable to attach itself to the receptor site of your cells. It cannot destroy your cells. Okay, next one, precipitation. Okay, what is precipitation? When you learn chemistry, I'm sure it's a break rate precipitate. Okay, that's for your glucose test. Precipitate is like in BM we call it mendakan. Mendakan is something that is solid. Okay, it's very it's not very liquidy. It's not like liquid. It's a very sticky thing, and it's like it will sink to the bottom. So it becomes like some sort of like solidified. All right. So precipitation. So antibodies react with the dissolved antigen, and then it will form precipitates. Okay, so things will become heavy. They might even drop to the bottom. So they form an insoluble complex. So you will see there are going to be granules or globules there, right? They are precipitates. And then now it makes it easy to be destroyed by phagocytes. So later again, the phagocytes will come and finish them off by eating them. So first, this thing is to immobilize them, right, and make them become precipitates. And then now your phagocytes will come and just eat them up. Okay. Next, opsonization. Okay, this may be something new to you, opsonization. So it's just it's a very fancy word, right? But actually it means very simple thing. It's just like marking it. Okay, so there are some antibodies that function. It doesn't really kill off the thing. It doesn't really sort of like break it down, whatever. It just marks it, marks the antigen, and act as a marker. So when you mark it, then it's like easier for the phagocytes to come and destroy them. So they recognize the antigen. Okay, it's already been marked. So it's easier for these phagocytes to just finish them off. Okay, by eating them. So this is opsonization. Okay, so that antibody is called opsonin. Opsonin is the antibody that is able to mark it. And then you have this phagocyte. This is the phagocyte that comes later and ingests the whole thing. Eats up the whole thing. Okay, and lastly, lysis. I'm sure lysis is simple for you. You've seen many things like hemolysis. Okay, lysis basically just means breaking down. Okay, bursting, broken. So antibodies will combine with this antigen. So this antibody is known as a lysine. Lysin. Lysine. Lysine is a type of antibody that can do this job of breaking down the thing. So lysine will combine with antigen, and what it does is, okay, so lysine antibody combined with antigen, and you can see the action here. The bacteria undergoes lysis. That means undergoes bursting and breaking down. It bursts, right? It just splits up, and this causes the bacteria to be broken down and decompose. Okay, so these five, what do you call this reaction? Action of the antigen antibody actions are simple to remember. So you just need to know, looking at the picture lah. So sometimes how you need to recognize is, if you have a picture, ask yourself, does this look like they're stuck together? If they're stuck together, that would be agglutination. And look at the diagram again. Does it look like the cell is broken? If the cell is broken, the bacteria is broken, then this is a lysis. reaction. Okay, so basically this is about it lah. All right, okay, so I have gone through what I have wanted to tell you today. Okay, so it's a simple lesson. Hope to continue next week. So if you have any questions, you can message me later. All right, and I will give you my Telegram name here. If you do have any questions, I'll be most happy to answer you. Okay, so thank you very much for watching, and I hope to see you again. Okay, just share my link to other people, your friends who might want to have some understanding of the chapter. Okay, bye-bye, everyone. See you next week. Bye-bye. Thank you, everyone. Thanks, Mohammed and Kenneth.